It's going to be a pre-recording. One of the invited speaker, Professor Violeta Oro, Institute for Plant Protection and Environment from Serbia. Application of scanning electron microscopy reveals the nematode microbiota. Professor, do you hear me?
Yes, I can hear you. I hope you hear me also.
Yes, I can hear you. Welcome again. If you are ready, I can start your presentation. Professor?
Yes.
Are you ready?
Yes, I'm ready.
Okay, I am going to start your presentation. And also, I want to say again, I would like to remind that this virtual congress is recording.
Dear colleagues, welcome to the 7th International Congress on Microscopy and Spectroscopy. Today's presentation will be about scanning electron microscopy and nematode microbiota. The invention of a microscope was an immense step forward for science, enabling development of many different disciplines. In the past, the living organisms were known only if they were larger than 1 square millimeter, and objects were in micrometers and could be seen only in micrometers. Nowadays, an electron microscope with its powerful magnification and resolution allows observation of organisms or their parts in nanometers. But how did it all start? Let me remind you on some milestones from the history of microscope. In the 7th century BC, Nimrod lens. It was a piece of rock crystal found in the Assyrian palace of Nimrod in today's Iraq. Much later, in the 10th century CE, reading stone was used as a glass sphere placed on top of text to magnify it. In 1021, Hightown wrote the Book of Optics, Thesaurus Opticae, describing light and vision. In 1284, first eyeglasses were constructed by Salvino Bernate. In the 16th century, Zachariah Janssen and his son Hans placed multiple lenses in a tube and developed an early microscope which used a visible light. In the 17th century, Galileo Galilei developed a compound microscope with one convex and one concave lens. In 1625, the first use of term microscope by Giovanni Faber. 40 years later, the first use of term cells by Robert Hooke, who published Micrographia, describing cells of cork tissue. In 1676, living cells were first seen by Antony van Leeuwenhoek, who built a simple microscope with one lens to examine blood, yeast, and insects. In the 19th century, 1830, Joseph Lister reduces spherical aberration by using several weak lenses together at certain distances to give good magnification without blurring the image. In 1931, Ernst Ruska and Max Knoll designed and built the first transmission electron microscope based on an idea of Leo Szilard. 1942, Ernst Ruska constructs scanning electron microscope which transmits a beam of electrons across the surface of specimen. So the visible light was replaced by a beam of electrons. In 1986, the Nobel Prize was awarded to Ernst Ruska for the electron microscope and to Gerd Beining and Henry Guerre for the scanning tunneling microscope. In the period between 1993 and 1996, super-resolution microscopy developed by Stephen Hill. In the 21st century, 2010, atoms were seen for the first time by researchers at University of California, Los Angeles, using a cryo-electron microscope to see the atoms of aquariovirus. 2014, the Nobel Prize in Chemistry for super microscopes awarded to Eric Bessick, Stephen Hill, and William Warner for the development of super resolved fluorescence microscopy, which allows microscopy of the object smaller than 200 nanometers. Without microscope, we wouldn't be able to see the micro world of living organisms and microbiology, bacteriology, mycology, virology, nematology, and many other disciplines wouldn't exist. Nematology is the science of nematodes. Nematodes are microscopic invertebrates of varying dimensions. Some extremes are between 100 micrometers and 10 meters, but mostly there are a couple of hundreds micrometers. They inhabit terrestrial and aquatic areas and can be found as parasites on or inside other organisms. Electron microscope enables research on such tiny organisms and even organisms smaller than them, their parasites. While transmission electron microscopy demands small and thin cross-sections of samples cut with microtome, in scanning electron microscopy, samples should be conductive enough, enabling the electron beam to scan the surface. Cis nematodes are an important group of plant parasites causing economic loss of millions of dollars annually. Potato cis nematodes, Globodera pallida and Globodera rostoviensis, are the most famous cis nematodes, and they are parasites of potato, tomato, peppers, and some other solanaceous plants. Females have spherical bodies called cis attached to the potato roots. These white spherical bodies are actually females attached as parasites or on a potato root in the background. Cis dimensions vary between 100 and 600 micrometers. And this is a scanning electron micrograph of Globodera cyst with its dimensions. Cysts remain in soil for a long time, enabling various microorganisms to colonize their internal and external side, forming its microbiota. Sometimes some microorganisms are deleterious and cause cyst death and decline of population, acting as biocontrol agents. This research demonstrates the application of scanning electron microscopy in the studies of potato cis nematodes, enabling insight into their microbiota. Microbiota, in a broader sense, are all microscopic organisms associated with an eukaryotic organism. It can be bacteria, fungi, virus, mites, and so on. Bacteria are ubiquitous organisms inhabiting even the most extreme environments like polar snow, volcanoes, and acidic hot springs. And how many bacteria can be found in cysts? Cysts can contain 260,000 bacteria, but it depends on specific conditions. In this study, bacterial isolates from Globodera cysts were identified by molecular analysis with help of 16S primers. And what kind of bacteria we found in cysts? The majority belong to the class Bacilli, represented with the families Bacillus and Penibacillus. Another group that was found were actinobacteria with the family Micrococcaceae and Arthrobacter species. And we found also alpha proteobacteria represented with the family Hypomicrobiaceae and Evoxia species. This is micrograph of bacillus species. Apart from bacteria, we found fungi also. Fungal isolates were identified by morphodimensional analysis using slide cultures. Only Sterile mycelium was identified by molecular analysis using ITS primers. And we found great taxonomic diversity of isolated fungi. They belong to different phyla, Ascomycota and Basidiomycota as well. The members of Ascomycota were Fusarium, Botrytis, Aspergillus, and Geotrichum, and one representative of Basidiomycota, Oxysporum species. Fusarium Oxysporum is well-known plant pathogen, belongs to the family Nectriaceae, Ascomycota, and it was isolated from Globodera pallida cysts. The species produces some toxins, such as fumonisins and trichotisins. This is a micrograph of a cyst, Globodera cyst, trapped by Fusarium Oxysporum. And this is the part of mycelium. Oxyporus lacte marginatus is the member of the family Polyporaceae, Basidiomycota, and the species was isolated from Globodera pallida X, so it was inside the cyst. In nature, this species is frequently found in association with beech trees and oak trees, Fagus and Quercus species. And it produces toxin, which is also antimycotic, benzylfuraldehyde. This is a micrograph of Globodera cysts trapped by Oxyporus mat and sterile mycelium. Botrytis cinerea is the next fungi, the member of the family Sclerosiniaceae, Ascomycota, and the species was isolated from Globodera pallida X, also inside the cyst. The species produces lytic enzymes and some toxins. Micrograph of Globodera cyst in Botrytis net and the part of mycelium. Aspergillus fumigatus is widespread fungi, belongs to the family Trichocomaceae, Ascomycota, and this species produces cytotoxic gliotoxin. and also some antibiotics, such as fustian, ferricrocin, fumigacin. This is the start of colonization of Globodera cyst by Aspergillus and its comedian. Geotrichum candidum is the member of Dipodascaceae family. Previously, it belonged to the yeast family, Saccharomycetes ascomicota. The species was isolated from Globodera lucifiensis cyst, and it was found to be parasite of the root knot nematode Meloidogene incognita. Pictograph showing start of colonization of Globodera cysts and the part of mycelium. It is clear that scanning electron microscopy is a superior technique in comparison with other techniques, in the sense of the most powerful resolution and magnification, but also the most expensive and laborious technique. There are great differences between specimens seen under the optical microscope. This is the reproductive area of Globodera cyst seen under optical microscope. And under the scanning electron microscope. Here we have that three-dimensional appearance, which is the closest to the natural look. Apart from fungi and bacteria, we found, unexpectedly, scanning electron microscopy revealed the presence of organisms not detected by the optical microscope. At first sight, I thought there are artifacts, many impurities on the cyst surface, but with larger magnification, a completely new organism appeared, an unknown organism with strange morphology and with very strong mouth, strong jaws, and legs. This microscopic invertebrate should be included in the list of natural biocontrol agents. So we can conclude that scanning electron microscopy revealed the presence of unknown organisms that are often overseen by standard techniques. Thank you.
Thank you, Professor, for your nice presentation.